Wishing you all a lovely morning. In our previous class, we had the quick revision of chapter 10 cell cycle and cell division. So today we will continue the cell cycle and cell division. So in our previous class, we have seen what is cell cycle, mitosis, the various stages of mitosis, significance of mitosis. Today let us learn meiosis, the various stages that is taking place in the meiosis and its significance. So what is meiosis actually? Meiosis is a very important topic. In meiosis, double division is taking place. For the gamete formation only, the meiosis is taking place. Meiosis is otherwise called as reductional division. Why it is called as reductional division? Because from the parent cell, when it goes to the daughter cell, the chromosomes becomes reduced. That's what it is termed as reductional division. Okay, meiosis is otherwise known as reductional division. So, in meiosis, it is being, chromosome number is being reduced. Just it becomes half. That's what it is known as reductional division. So, meiosis will be taking place in the reproductive cell. Only in the diploid cells it will be taking place. So, 4 cells are generally produced in this stage. And also, after that, the variations will be introduced in the meiosis. How the variation is being introduced, how variation is taking place because of the gene exchange. That's what variation is being introduced. Now meiosis, we can divide them into 2 parts. What are they? Meiosis 1 and meiosis 2. Actually in the meiosis 1, the actual meiosis is taking place. In meiosis 2, actually the mitosis is taking place. So let us see the meiosis 1 and its different stages. So what are the various stages of meiosis 1? You can see here. So the various stages of the meiosis 1, prophase 1, metaphase 1, anaphase 1 and telophase 1. So these are the various stages. So prophase 1, that is the longest phase, and we can say that it is a complex phase. The longest phase and the complex phase is the prophase 1. So prophase 1 has to undergo various stages. What are they? They are leptotin stage, zygotin stage, patatin stage, diplotin stage and diakinesis. So these are the various stages that which is under prophase 1 is undergoing. So let us see each one in details. So the first one is of leptotin stage. In leptotin stage, the condensed chromosomes will be condensed. Okay, they will be condensed. And the structure of the arrangement, as we have seen in the mitosis, they will be like a bouquet type of arrangement in the leptotin stage. Chromosomes will be condensed and the structure will be of bouquet type of arrangement. And nuclear membrane starts to disintegrate. Okay, it starts to break. Nuclear membrane starts to disintegrate, or it starts to break. And next in the zygotin stage. Zygotin stage, what is happening? As I have told you, 5 stages are there. First stage is over. And in the second stage, zygotin stage, the homologous chromosome starts to pair. The homologous chromosome starts to pair. So that pairing is termed as synapsis. The homologous chromosome starts to pair, and thus that chromosomes are termed as synapsis. And here in the zygotin stage, we can see the bivalent appearance, or we can say tetrad type of appearance. So it looks like 4. So it is called as the tetrad like of, or bivalent appearance is termed as synaptonomal complex. So it is termed as synaptonomal complex. So this will be mixed. Okay, synaptonomal complex, they will be pairing. And from which stage it is being started, synaptonomal complex? It is starting in the zygotin stage. So first in the leptotin stage we have learnt. Now zygotin stage is over. Next let us move to the patatin stage. So in patatin stage, what is happening? Tetrads, this pairing of the chromosomes, it become more visible. We can say this is the longest stage. And we can see here in this stage, patatin stage, we can see the recombination nodules. We can see the recombination nodules. So here in the recombination nodules, what is happening? Crossing over will be taking place, as we have seen in the case of mitosis. So homologous chromosomes will be, the pairing will be taking place. So it forms non-sister chromatids. So for this, what we call it as crossing over. So crossing over, the homologous chromosomes जो सेगमेंट के बीच में जो ओवरलैपिंग हो रहा है उसी को क्रॉसिंग ओवर बोलते हैं. क्रोसिंग होमोलोगस क्रोमोसोम के ऊपर जो ओवरलैपिंग हो रहा है उसी को क्या बोलते हैं? क्रॉसिंग ओवर बोलते हैं. सो दिस इज व्हाट इज हैपनिंग इन द पैचिटिन स्टेज. सो इन द पैचिटिन स्टेज tetrads are more visible. We can call it as the longest stage, and recombination nodules are being taking place here. Next is of diplotin stage. In diplotin stage, what is happening? As we have learned in the zygotin stage, synaptonemal complex, it starts to dissolute. Okay, so synaptonemal का dissolution होगा diplotin stage में. Then bivalent, this tetrad will be disappear and chiasmata formation will take place. What is chiasmata formation? X type of formation, that is chiasmata formation. So synaptonemal complex start to dissolute and bivalent disappear and chiasmata formation starts here in the diplotin stage. And in the last but not the least, in the diakinesis stage, what is happening? So we can call it as the end part of the prophase first. Okay, so chiasmata, the formation of the chiasmata that which is occurring in the diplotin stage will be terminalized here in the diakinesis stage. And also the mitotic spindle will be assembling here, nucleolus starts to disappear, nuclear envelope starts to break here in this stage. So I hope all these stages are clear to you all. First stage is leptotin. So just we will revise once again. Leptotin stage, you have to remember that replicated chromosomes condense and bouquet type of appearance, nucleolus start to disintegrate or break, we can say. Then the next stage is of zygotin stage. Zygotin stage, as I have told you, bivalent appearance plus synaptonemal complex, you have to remember. And in the patchitic stage, tetrads become more visible and crossing over takes place here. And in the diplotin stage, what is happening? Chiasmata formation is taking place, and in the diakinesis stage, terminalization of the chiasmata is taking place. So here, nucleolus starts to disappear, nuclear envelope starts to disappear, mitotic spindle starts to move in the poles. So these are the various stages of meiosis 1 and various stages of prophase 1. Now let us move towards the metaphase 1. Here, in the image, we can see the various stages of meiosis 1. The various stages of meiosis 1. So prophase 1, metaphase 1, anaphase 1 and telophase 1 we can see here. So in the prophase 1, the chromosomes are condensed here. And in the metaphase 1, you can see here, the chromosomes are arranged in an equatorial plane. So it is aligned. It is aligned in an equatorial plane in metaphase 1. In anaphase 1, what is happening? Anaphase 1, the chromatids will be associated. The homologous chromosome starts to separate. Here you can see the separation. It starts to separate while sister chromatids remain associated at their chromosomes. So these sister chromatids will not be breaking, they will be associated together, but they will be moving towards they will be separating. They will be moving towards the various poles. And in the telophase, what is happening, as I have told you, the nuclear membrane, the nucleolus, everything reappears. So that which was being disintegrated here in the telophase stage, it reappears. Nucleolus appears, endoplasmic reticulum appears, Golgi complex reappears. So reappearing is taking place in the telophase stage. And it will not move to the extremely extended stage. So now let us move towards the meiosis 2. So what is happening in the meiosis 2? We can call it as homotypic division, or we can call it as equational division. This meiosis 2, we can call it as homotypic division or equational division. Why? Because what we have seen in the mitosis, same thing is taking place here. Equational division, why do we call it as? So the chromosome number will be remaining same as in the parent cell to the daughter cell also. So meiosis 2 and mitosis, there is a small difference. What is the difference here? In meiosis 2 will be taking place in haploid cells, while mitosis will be taking place in the diploid cells. And one more difference is there. So due to the crossing over, the chromatids of the chromosomes will appear dissimilar, but in the mitosis it appears to be similar type. So that is the main two differences. So one difference is, as I have told you, meiosis will be taking place in haploid cells, while mitosis it is taking place in the diploid cells. And one more difference is that due to the crossing over, two chromatids of the chromosomes will be appearing as dissimilar size, dissimilar size means entirely different. But in the case of mitosis, the two chromatids of the chromosomes will be having a similar look. So this is the main difference. This we have already seen. So next, moving towards the prophase 1 stage. So prophase 2, in the meiosis 2, prophase 2. So what is happening in the prophase 2? So chromosomes have fully elongated. We can see here in the image, the chromosome has fully elongated. And in metaphase 2, chromosomes align at the equator, and in anaphase 2, what is happening? Splitting of the centromere is taking place. Splitting of the centromere is taking place. So we can see here in the image, it is clearly given. So in anaphase 2, splitting of the chromosome, here it is aligning, and here the chromosomes are being elongated in the prophase 2, as you can see here in the image. So I hope it is very clear to you all. Chromosome second, the chromosomes are being elongated. In the second part, metaphase 2, we can see that it is being coming in the aligned in the equator level. In anaphase 2, sister chromatids separate. And in the telophase 2, what is happening? Telophase 2, the meiosis, second meiosis ends at telophase 2. So here, in the telophase 2, second meiosis is being ended, and cytokinesis follows, resulting in the formation of tetrad cells. So what is forming here? 4 daughter cells are being formed. 4 haploid daughter cells are being formed in the meiosis second. So hope you might have understood. Now let us see the significance of meiosis. What are the significance of the meiosis? What is happening in the meiosis? So this is very, very important. Significance of meiosis is very, very important. Here, the formation of gametes is taking place here, and we all know that why it is called a reductional division. As I have told you, the chromosome number becomes half the number. And here, the chromosome number, the conservation of the specific chromosome number. So the chromosome number will be conserved. One of the main point of the significance of the meiosis, we can remember that conservation of the chromosome is being taking place here. And next is it increases the genetic variability. So variations are taking place. It increases the genetic variability. Where this in nature, where the variation occurs in the from one generation to next generation, when it moves, it occurs in the population. We all know that. So variation is very good. So variation is very good or very important process in the process of evolution. We know that. And the last and important point is that mutations. Mutations are also taking place here. So good mutations are there. Due to the mutations, what happens? New combinations arises. That's what I told you. That is taking place in the natural selection, and all. When we have learned, we may know that about the genetic mutation we might have learned. So good mutations will be taking place means, so new combinations may come. So here we have completed this chapter. Hope you might have understood what is cell cycle, cell division, and how the cell division is taking place in cells. Then what is the importance of mitosis, what is the importance of meiosis, the various stages of mitosis, the various stages of meiosis, and the significance of mitosis and the significance of meiosis. So please go through your NCERT text, please prepare a note according to it, so draw all the images which is necessary for your studies, and learn well. So happy learning to all of you. Thank you. May God bless you all.